thank you all for coming. I just want to start this evening's conversation with just a shared moment of silence. Thank you.
Thank you.
Again, good evening, everyone. Thank you for joining us. My name is Ava Della Santa. I am the Assistant Director of Regional Alumni Engagement. We are looking forward to a wonderful conversation this evening, and I would love to introduce our featured speaker. But before we get started, I, of course, have a couple housekeeping items. I want to encourage everyone to keep your video on if you're able to. That just helps us feel the participation of everyone who has joined, and virtually, we can still feel that sense of community. I also want to encourage you to change your Zoom name to include your full name and add your class year or your constituency if you're a current parent, grandparent, perhaps faculty, staff. And this just helps us kind of know who you are. It's your virtual name tag. And you can do this by clicking the three dots in the corner. And feel free to chat me if you're having any trouble. We do ask that you keep your microphone muted as much as you can, just to minimize disruptions. Of course, when we start asking questions and such, you can absolutely unmute your microphone. We have a little change of pace this evening that I just want to share because it does change my housekeeping speech a bit. William Howe is here, class of 1986, and was supposed to be in discussion with classmate David Shizgal. And unfortunately, he is not feeling well this evening and was not able to join us. So William has done a quick flip on tonight's conversation, and we're really excited for him to just share with everyone a nice slideshow and then also encourage a lot more audience participation. So this one's a little different. So stay muted if you're not speaking so that when you do speak, we can see who is speaking. And then also, there might be moments where he'll ask questions to the whole group. Please feel free to write answers in the chat. They'll come to myself, and I'll be happy to read them out loud and share. But yeah, I think we're ready to get started. So I guess I will just turn it over to William, and thank you so much for your flexibility this evening.
All right. Thanks a lot, Ava. Thanks for hosting. I'm really excited to be here, and it's great to see so many familiar faces. Literally 10 minutes ago, I found out that David unfortunately couldn't join us. So if you guys have his email, send him a note and wish him well.
What?
So with that, I put together some slides on the fly. We'll have a chat and then we can open it up for Q&A. And we'll just keep it informal. And thanks again for joining. So let me see if I can share my slides here. Hold on okay, yeah. Give me 1 second. Okay, here we go. All right, can you guys see that? Yeah?
Yes.
Okay, all right. So basically, the only reason I'm showing this slide is to show the background in terms of drug development. I think you guys know after class of 86, I went to Harvard where I majored in East Asia Studies. Then I ended up at Yale where I did an MD PhD where I worked on the function of gamma delta T cells. And David was supposed to ask me about my background, so I had to add this slide here. Then I did internal medicine at Cornell here in New York, medical oncology fellowship at Memorial Sloan Kettering, where I worked on cancer genetics and lung cancer, doing research as a physician, also had a lab and saw patients. And then I was at Vanderbilt, where I eventually became the division chief. And then during that time, also had a lab, helped see patients and help run clinical trials. Particularly, I saw lung cancer patients. And then I got recruited to head up oncology drug discovery at Roche, based in Switzerland. I did that for 4 years, and then I was head of the whole unit there, which was immunology, infectious disease, oncology, neuroscience, rare disease, and oncology. And then more recently, I was the chief development officer at Pfizer. And then now I'm CEO of a biotech here in New York. So if you guys are in New York, I would love to catch up and see you guys. Anyway, during this time, then I've helped develop over 20 medicines that have been approved. So then I feel this is just as a background in terms of the context for why I'm writing a book about innovation in drug discovery. Okay, so some of you might have met my dad, but this is a really a young picture of him. And my book is dedicated to my dad, who unfortunately, I think you all know, he died when I was 13 at Sidwell. And Sidwell really was a family for me during that time. But unfortunately, he died of metastatic colon cancer. I don't know if you all know that. It was unexpected. 2 years before that, he had colon cancer was diagnosed, and then he got chemotherapy. Unfortunately, he lost his hair and got really nauseated and really sick. But then he recovered, and we thought he was doing okay. But then in 1981, he went for a biopsy and unfortunately didn't make it out of that and died of metastatic colon cancer. So at that time, I really did vow to make a difference for patients like himself. Didn't know how. You guys know both my parents were doctors, and then my sister's a doctor too. So sort of expected that everyone in the family would be a doctor, but I didn't know exactly how I would do that. And then after 20 years of training, basically, then finally was able through lab seeing patients and doing clinical trials for cancer patients, able to help patients like himself. And he really is a hero because basically he came from China in 1949, 1948 actually, and I think legend has it that he got paid $100 a month at the time when he was a resident. All right. Anyway, so since we're trying to keep it informal, how many molecules do you think have ever been approved in the history of the U.S. in terms of new medicines for patients? As of December 22. So a medicine or a molecule is supposed to improve how we feel, function, or survive. And the FDA was only started in the late 1900s, so sort of includes some medicines that may have been approved before that, technically approved. But how many do you guys think have been approved in the history of the US? 180, 1800, 3600, 10,800, or 18,000? Anyone?
D,
David,
600.
600,
18,000.
18,000. Okay. Okay, well, that's a couple answers. So the answer is 1,800 as of December 22. So I tend to see that as a very low number. You heard somebody say 18,000, so it might have been thought. I'm talking new molecular entity in the sense that it's like a new mechanism. It's not like the next iteration of Tylenol cold or something like that. So why is it so challenging, and how did these breakthroughs come about? So just some facts and figures about human beings. I think you guys might know there's 78 organs, 206 bones, but more importantly, there's 20,000 protein coding genes in the human genome. And then there's about 90,000 protein coding RNA transcripts. And remember that it goes from DNA to RNA to protein in the central dogma of biology. On top of that, so human beings have 30 to 40 trillion cells. And each of these contains their own signaling pathways, their own molecules, their own structures, their own biology that we still don't fully understand. And then on top of that, there's 8 billion people on the planet. So you can imagine having to make a medicine that would be well tolerated and benefit, maybe almost 8 billion people on the planet becomes very challenging, particularly because from a safety point of view. Unlike mice, which are genetically engineered to be all the same, obviously human beings are all very different. And so once you even get into human beings, how they react to different medicines and stuff can be totally different. Okay, so do some of you know who Art Levinson is? So Art Levinson was the CEO of Genentech, which is a famous biotech. He was also like CEO of the year several times. He's currently chairman of the board of Apple, actually. Quite a remarkable guy. But I used to talk to him about drug discovery when I was at Roche, because he was previously at Genentech, which was bought by Roche. And he's like, yeah, drug discovery is not chess. And I thought about it, and chess is pretty complicated already, where there's 10 to the 40th possible moves, but we know all the pieces on the board. We know what each piece can do. We can move one piece at a time, and there's a finite, basically, board. But in drug discovery, we don't know all the possible moves. We don't know all the pieces. We don't know what each piece can do. All the pieces are moving all the time, and then we don't even know the extent of the battlefield. And for every rule that you have, you always have an exception. So that's why some of us say biology is not physics. It's not like force equals mass times acceleration or distance equals rate times time. For every rule, there's always a exception, which then makes science hard to progress forward. All right. So when you think of innovation, or at least I think when the public thinks of innovation, they think about books like from I'm blanking on the guy's name, Walter Isaacson, who wrote about Leonardo da Vinci. You read about going to the moon, you read about computers and telecommunications, or you read about Tesla and electric cars, but why not drug discovery? So I thought particularly, there's just as much, if not more innovation going on in drug discovery because you actually have to put drugs inside yourself. And they can't kill you and they're supposed to help you, unlike, an iPhone, which you can put in your pocket, but you don't put in your body. And then you're also trying to improve upon billions of years of evolution if you believe in evolution. Okay, so why did I write Breakthrough? Basically for three main reasons. The first was I really wanted to highlight the unsung heroes of drug development. So most of the time in science, like a Nobel Prize or these prizes or drug discovery, you hear about one person. And then also when a new medicine is launched from a company, the company just says company X launched this medicine and it doesn't say anything about the scientists behind it. But the truth is there's thousands of people working hard every day to try to develop new medicines. And on the right in the circle there are just some of the different kinds of roles, which I didn't even know about when I was in academia and went into industry, of all the subspecialties and different people that can work on or have to work on a molecule and getting it over the finish line. These include not only biologists, chemists, protein engineers, toxicologists, clinicians, and stuff, but really a lot of other roles. At one point, we counted up the number of people who worked on a molecule at Roche, and it was 3,000 who ultimately had to get that molecule over the finish line. The second reason is, there's too few books about innovation in drug discovery. I just mentioned there's books about going to the moon and other places, but there's really not a lot about drug discovery. And then finally, I wanted to inspire the next generation of scientists and innovators, whether they be at Sidwell or at other places. And it's not only to innovate in life sciences, but other areas as well. Okay, so basically the book talks about 8 different pioneering medicines from 8 different companies. Some of these may not roll off the tip of your tongue, but they're all really cool medicines, at least to me, including risdiplam, which is the first oral RNA splicing modifier. That's a full mouthful. I'll explain a little bit about that in more detail. Osimertinib, which is a drug that I actually helped develop from academia, which is a type of oral medicine for lung cancer. Then paracetamol, Tylenol, you guys all probably know. That's the first anti-fever medicine, which was actually accidentally discovered. That's a very interesting story where basically there was a guy with intestinal worms in Strasbourg, and he went to see some local doctors who prescribed to him naphthalene, which is the chemical used for mothballs, the smell that you see from mothballs. That was at the time in the 1890s what people used. Anyway, he went to the pharmacy, he got a prescription. He took the medicine. His intestinal worms didn't go away, but he had a fever, and his fever actually went away. And when he went back to tell the doctors, they're like, "What the heck?" Because they had never seen a medicine treat fever before. I mean, now we take it for granted, but back in the 1890s, like that was not the case. Anyway, so then they went to the pharmacy to make sure to see exactly what the patient got, and it turned out the pharmacist had misprescribed the medicine. It wasn't even naphthalene. Like today, that would like get that pharmacist in jail for malpractice. But at the time, it ended up being a novel discovery because the 2 doctors said, what did you give him? They figured out what the medicine was. And then it turned out to be, they developed what's called antifebrin, which was the first anti-fever medicine. So the whole story about how the discovery of Tylenol was made was pretty crazy and pretty accidental. Then there's a medicine called emicizumab for hemophilia A. Alpelisib for a type of breast cancer. Paxlovid is in there, which was quite a remarkable discovery, the antiviral for COVID in less than 2 years. Then there's the first medicine, which is a gene editing medicine for sickle cell disease. And then there's a molecule called lenacapavir for HIV. It was actually named Science Magazine's 2024 Breakthrough of the Year last year. I did not know that it was going to be named that when I started writing the book. But essentially, all of these medicines are really cutting edge in terms of the science, except for paracetamol. And they all have some kind of story where there's a resilience, a really required a lot of breakthrough, and it required decades of work in order to finally get these medicines approved. Okay, so this is a disclaimer about the book. It's really about innovation, about drug discovery and development, and it's highlights the stories of the science and the scientists behind the different molecules, but it's not an advertisement for pharma. It's not sponsored or written by pharma. And if you want to know anything about the medicines, if you want to use them for yourselves or for your personal family members, you have to talk to your doctor or look at the prescribing information. Okay. So I have to say this is a little awkward speaking in front of my Sidwell colleagues about this. I was expecting to do more of a fireside chat with David, but that's okay. So you guys feel free to ask any questions or if you have any comments, just feel free to interject. So with that, then I'm going to talk a little bit about one of the molecules that treats a disease called spinal muscular atrophy. This is one of the most common rare diseases, which affects young babies. You can see the frequency up there. The most severe form of this is called floppy baby syndrome, because basically babies are born and then they are unable to control their muscles because they have trouble innervating the muscles from the neurons. And then they become floppy, and so they can't even sit up. And so some of them, actually, the most severe form, they die by the age of 2. They also have difficulty breathing and so on. And it's really a disease of the degeneration of motor neurons. So in the past, there are now 3 new medicines for it, but even a decade ago, there was no treatment for this disease. And it's an autosomal recessive disease. Those of you guys who had Mr. Biggs would remember what an autosomal recessive disease is. So basically, you have to have 2 parents, one of which is a carrier, the other which is a carrier, and then if both of them are carriers, then 1 in 4 chance of getting the disease. Okay, so this is the most complicated science slide for the audience, but just bear with me for a second. The book is written for a lay audience, but I do have to explain some of the science that goes into the making of the molecules. So basically, just remember on the left, the central dogma is you go from DNA to RNA to protein. DNA is like the blueprint. RNA is then what the plans are made to ultimately make the protein. And then the protein is what affects the molecules inside the cells. So it turns out that DNA, as you can see on the top right, has what are called introns and exons. So exons are the boxes in the top here, and then introns are sort of DNA in between. And the exons are actually what encodes the genes. And during the process of going from DNA to RNA to protein, you actually have to splice out the introns, and then you can see that the exons form together. And then ultimately, it's the exon that's then translated into proteins. The reason this is important is because risdiplam is an RNA splicing modifier, which then is miraculously differentially affecting how exons are spliced in humans once people take it. So now this is a little bit more complicated too, and then I'll get less sciencey. So in patients, in normal individuals on the left side, we all have 2 genes. We have one called SMN1 and SMN2. This stands for survival of motor neuron 1 and 2. And basically in SMN1, there's this exon 6, there's exon 6, 7, and 8. Exon 7 is spliced properly, and then you have an mRNA that has 6, 7, and 8, and then you make a stable protein. SMN2 has a mutation right here, and therefore exon 7 is not included in the RNA. You see here you only have 6 and 8, and 6 and 8 translates to an unstable protein. So what this means is that SMN2 can't compensate for SMN1 normally. Now, in the middle is a patient with SMA, and they actually have a defect in SMN1. So they can no longer make the protein. And as I just said, SMN2 can't compensate. So you can imagine like a car with two headlights and one of them is broken. And then can you actually make the other headlight compensate for the broken one? That's the analogy here. And when you take a medicine that we developed, this risdiplam molecule, it actually tricks the cell into including exon 7 and then having the RNA have 6, 7, and 8 so that patients can make a stable protein. So that's the most complicated I'm going to get, but I intentionally described that because the level of science of some of the molecules that are being developed now is really sophisticated in terms of the understanding of what has to be done, and then how do you fix it. Now, in terms of the public, the way that drug development is usually depicted is as a linear process. So basically, there's two main phases. One's called a preclinical stage and one's a clinical stage. And then in the preclinical stage, you have all these different steps. And then ultimately, you get into clinic and you start testing your molecule in humans. So it's nice to depict it in a linear way. But in truth, it's really not a linear process at all. So now if we go back to the discovery of risdiplam, which is this molecule actually that came out of my group at Roche PRED, it turns out it was basically 100 years in the making. And that's part of the point of the book, which is like these medicines don't appear out of thin air. There's like decades and decades of work that has to be done. And I don't want to be too controversial here, but some of the stuff that's happening in the NIH and stuff right now is going to really affect the future of innovation in terms of being able to build upon scientific knowledge over decades. It is really important to fund basic science research. So if you look at risdiplam, then the disease was originally SMA was originally described in the 1890s. Then DNA wasn't described till the 1950s. RNA splicing was discovered in the 1970s. In the 1990s, Judith Melke in France was the first to discover the genes involved in spinal muscular atrophy. Then Adrian Craner at the Cold Spring Harbor showed a decade later that you could actually splice SMN2 and have it compensate for SMN1. And then finally, with all of this knowledge, 3 molecules were approved in the 2010s and 2020. And so, again, this makes it look linear, but really drug development is not a linear process. So one of the key important things is that there's a triad in drug discovery, and I call this the understanding of biology, the clinic, and technology. So you need to have a cutting edge of understanding of disease biology. You need to understand the disease and exactly how the biology relates to the disease. And then you need to have a drug basically that can hit whatever target you want to hit. And only when you get to the intersection of all 3 of these could you actually get a launchable drug. And that is really the crux of drug development, as opposed to just doing biology for itself, clinical stuff, or just technology on its own. And just to show you how complicated drug development is, then if we look at the example of spinal muscular atrophy, it's not linear again. Like, so first the disease was described in the 1890s. Then you had all these biological discoveries in the 1970s and so on. And then you had technologies being developed over here. And only when all of these 3 things came together, then all of a sudden you had 3 new medicines approved within the span of 4 years. And that's just how innovation works, right? People are working all the time trying to make improvements, and then all of a sudden everything comes together and you can make some big breakthroughs. So so basically in the book, once you go through the 8 chapters, then there's a final chapter on 10 insights from the front line of drug development. I talked to you about getting to the Holy Grail, which is the sweet spot of biological understanding, clinical understanding, and technological advancement. But some of the other themes are, 1, definitely innovation requires determination and resilience. And each story has basically almost every molecule was killed during development. But resilient scientists and clinicians and others really stuck with it and went through a lot of lows to finally get to approvals. And you have to read the book for the other 8. But they are applicable to other areas outside of life sciences, and I think in whatever field you want to innovate. So basically then, I would ask you guys, next time you take a pill, to take a moment and think about, who are the unsung heroes to who actually made that become a reality, how many people contributed to that and over what time frame, what obstacles had to be overcome, and then really celebrate the triumph of human ingenuity. And I would add further, it's scientists that come up with these breakthroughs. And so we need to really be supporting our scientists during this interesting time. So with that, I do want to thank my publisher, One World, my agent, Ewan Thornycraft, the multiple companies who agreed to have me interview their scientists. I interviewed more than 100 people. And then Victoria Powell, who supported me and actually gave me a lot of feedback during the writing of the book. And with that, I will stop and take any questions, if there's still anyone in the audience. I can't see anybody. Okay, there we go.
I got a question, William.
Okay.
So on that triad, I'm wondering if there's really at least one more big circle, right? And that's money,
Yes.
especially today, right? And it's not only the existence of money, but it's the potential of money in the future, right? That's why you don't. So how have you seen that change from, the innovative compounds from the early days to today?
Yeah, so you're talking about, David, you're talking about several things. One is commercial opportunity, right? Unfortunately, drug development is a business. And so obviously, pharma gets a bad name for drug pricing and a lot of things, but it is expensive to develop a drug. And for this audience, I'll tell you, here's just the math, right? So basically, at Roche period, I had $1.2 billion a year. And my job was to hand over molecules to late stage on average 10 over 5 years, which means about 2 molecules per year. So you can imagine then that if it's 1.2 billion a year, then 600 million per molecule that you're handing over to late stage. Now, the challenge is what I didn't talk about is the failure rate of drug discovery. So for every molecule that you bring into clinic, actually the failure rate is 95% or so. So the 600 million there per molecule is also taking into account a lot of the molecules that failed. And then also all the iterations, so even if you bring in a molecule for the first time and it's successful, they might have some toxicity issue or some other thing that you have to go back to the drawing board and fix it and so on and so on. So that's just to say that, drug development is expensive, unfortunately. But I do think it can get cheaper over time. And as we get more knowledge and move faster and we know how to do things better, hopefully it can get cheaper and cheaper, particularly for rare disease populations, smaller and smaller patient populations. That's probably not a satisfactory answer for you, but that's the reality.
Yeah. Well, I guess I'm wondering, were the early folks, the folks who were who found the first fever reducer, right? They weren't thinking, well, I'm going to make $1 billion out of this, right?
No. Yeah.
But to get something new today, you either have to say, this is my life's passion because I know so-and-so who's one of the 10 kids in the world who has this, and I don't care if I'm making money or not. Or you're saying, I'm going to make a Me Too drug and it's going to pay for my yacht, right? So, but where's that intersection right now where you're seeing money put in, a lot of money put in just in the name of making people healthier?
Yeah, so I think funding for science comes in multiple forms, right? First of all, a lot of the basic science research is done through NIH. And when I say basic science, it means asking fundamental questions like, why do cells grow? It's questions that are not necessarily going to lead to a drug. And you'll see also in the book, there's many scientists who were just working on pure curiosity questions who were not having anything in mind about making a drug. In fact, the guy who won the Nobel Prize for one of the discoveries predicted in 1950, he didn't even know that you could make a drug, which would be 50 years later kind of thing, right? So one is that fundamental basic science research is really, really important because also you have no idea when some of those ideas will ever even come to fruition, and probably many of them won't even come to fruition, but they'll be answering good basic nature's questions and stuff like that. Then I think what you're talking about is right now there's a lot of money in the venture capital world. I think there's some people in the audience here who might be doing some of that. where, right now I'm a biotech head also, and, I have to get funding Series A for my biotech. I have to have an idea, I have to convince people that, the idea is worth it. And also that there's a potential commercial, commercial potential down the line and so on. But the venture capital funds do fund a lot of the research now that lead to major breakthroughs in drug discovery. I think now more than 60, 70% of drugs that eventually come out of pharma originated in biotech.
Hey, William, I got a quick question for you.
Yeah.
I'm always amazed when a drug gets shot down for clinical trial design failure. How common is that? How hard is it when you're doing these clinical trials to know that the trial design is what the FDA wants? Like, whose fault is it when a trial fails for clinical design failure?
Well, I guess you could say partially, the people who designed it, but there's a lot of moving parts. So, when you're developing a drug, you also have to anticipate what will be the standard of care down the line. So basically, like let's say in 2025, if we're developing a molecule right now in phase one, which is the earliest stage, it may not get approved till 2035. So whoever's working on it has to anticipate, like, oh my God, in 2035, when it gets approved, like, is it going to be useful? How's it going to compare against the standard of care? Again, sometimes things that you don't even know are coming out. So I'll give the benefit of the doubt and say sometimes those failures happen because, the standard of care changes and then they're no longer, the molecule is no longer relevant. Oftentimes what happens, one of the criticisms is like you don't have the right control arm. Right. So in a phase 3 trial, you have to randomize people to a new treatment versus a control standard of care arm. And if the molecule is approved and the standard of care is changed, then it's totally irrelevant. And then you have to go back to the drawing board.
But you don't know that after you're spending all hundreds of millions of dollars enrolling patients.
You should.
Okay. Let me ask you one more quick question.
The other thing is, a lot of times I've seen in pharmas, you actually do those assessments. And then even if the molecule has been like successful up to that point, you have to just say, we can't invest in this further. It's really devastating. But you say like, this is not going to make it commercially. So I have to stop.
Okay. One more question, then I'll stop. But what are your couple thoughts on drug launch? Like drugs approved, it's another drug in a very crowded indication. I always wonder, like, are these doctors in their offices really able to analyze all the clinical data that's being thrown at them? How does a drug really launch, in your opinion? Is it salesmanship? Is it data?
Well, I think there's two classes of drug, and I would prefer to make breakthrough drugs, and they sort of sell themselves, meaning that like if a drug is really amazing and there's no precedence for it, you don't really have to do a lot of marketing for it because, for example, risdiplam, there's really not that many options. And the other therapies are very onerous. One is what's called an antisense oligonucleotide therapy that you have to get injections multiple times in the spinal cord. And then the other one's a gene therapy, which, may or may not work for all patients and stuff versus an oral pill that you take every day, right? So in that instance, then the data sort of speaks for itself. And then it's very easy to get people to be excited about it. Not only physicians, but also patients and stuff like that.
That's a monopoly. That's nice.
Okay, well, but anyway, then the other situation, if you really have to differentiate in a crowded market, unfortunately, I would say that marketing does make a difference there. I personally do have to say I agree that I don't know why we need pharma marketing. There's like billions of dollars spent on marketing of drugs, which I think is a waste of time. In Europe, where I lived for 8 years, there's no pharma advertising. So when you watch TV, you do not have to sit through a gazillion ads on like to thyroid eye disease or like, like some rare disease that you don't even know about. So I do think that is a waste of time. And if we save money on that, maybe we can make a lot more breakthrough drugs.
William,
Go ahead, Julie.
can I ask what areas, this is so fascinating, and thank you so much for doing this. What areas are you most excited about in the next 5 to 10 years? Areas of disease or science that you see breakthroughs coming or you're most excited about?
Yeah, so in the last chapter in the afterword, I talked about how this is the biological century. So, now we really understand a lot more about human disease, about biology. We have the ability to make new technologies much, much faster. And then we know how to develop drugs now a lot faster. So I do think there's going to be a lot of more breakthroughs, whether they come from the US or from other places, that's a second question. But I think we're going to have a lot more breakthroughs coming. Right now, the hottest areas are in what's called cardiovascular metabolism. Particularly, you guys probably know about GLP-1 agonists and these medicines that are being used for obesity and diabetes. There's a lot of investment, a lot of breakthroughs there. I do think there's going to be more breakthroughs coming in Alzheimer's disease, Parkinson's disease, and other areas. And oncology still, unfortunately, remains a huge area of unmet medical need. The number of cases of patients dying from cancer is expected to double by 2050, almost, which is sad across the world. But yeah, there's going to be new breakthroughs. There's actually some breakthroughs coming, I think, even in hearing loss from gene therapy, maybe in blindness through gene therapy. So stay tuned. There's going to be a lot of breakthroughs, I think.
William, you mentioned cuts. By the way, I downloaded the book while you were presenting it, so I'm looking forward to reading it tomorrow. You talked about like cuts to NIH. I mean, are there other areas of federal funding that are at risk right now that could affect the industry and could affect the research side and development of drugs? Where are the danger zones?
Well, I don't want to get too much into it, but there's a lot of budget cuts happening all over the place. In particular, the NIH, there have been grants frozen. There's this whole debate now about indirect costs. I don't know how much detail you guys know about that, but basically, the universities stand to lose a lot of money. And I do have a lot of colleagues in academia who are, laying off people. There's universities that are not hiring, not taking on new graduate students and things. So this really going to affect the future, I think, of science and drug development. The other thing from the FDA is, there's been a lot of cuts in the FDA. And so there is a concern. I just signed a letter with biotech executives to try to restore a lot of those people because into the FDA because we could really see a slowing down of approvals. And then the CDC is getting cut and so on and so on. I personally don't think this is a political thing. I think it's really about the health and innovation of the US. And I would argue that, the center of innovation for life sciences does not always have to be, it's not a given that it would stay in the US.
William, could you kind of dovetailing onto that, could you speak a little bit about what you think the role of AI will play in the development of drugs? And also, as a second part of that question, do you think there's going to be an acceleration of discovery of cures for like the more rare diseases because of AI?
Yeah, so Kira, that's a great question. AI is being touted in all parts of society, but also in drug development. There's a lot of hype, I would say. But I think it's definitely helping already in certain areas. The hype part is, I think the question some of us have is, can it lead to actually new serendipitous discoveries? So you'll see in the book, and a lot of science happens through serendipity. And that's through like somebody recognizing that like, oh my God, this is a totally wrong or unexpected result, but actually it turns out to be really important. And I don't know if a computer can do that because usually AI is based on what is previously known. So we'll have to see. I used to say that I didn't think that it would win a Nobel Prize, but unfortunately AI just won a Nobel Prize last year. There's a program called AlphaFold, which is a program developed by some, I think, ex-Google folks who have figured out how to model protein structures based on known protein structures. So anyway, it's transforming the field, but the point still there is for an unknown protein structure, I don't think it can visualize what that looks like. It can visualize what's known already. Anyway, the point is, it is already helping. You can, if you wanted to like go after a target and you had 3 million molecules that you wanted to screen, potentially AI could narrow it down to like, 1,000 or 10,000. It can help you with data gathering much faster. It could potentially help identify patients faster around the globe, to enroll for trials and so on. So I do think there are areas where it will help, but there are also other areas, hopefully, where the human brain is still required.
I guess my question with that is if you were to use it as a tool to connect all the human beings, because I totally hear what you're saying, and that's really cool to understand the serendipity of it all, which makes a lot of sense. But if AI is used more as a tool rather than the brain,
Yeah.
to connect people from all over the world and find these random cases that actually turn out to be related, is that an effective way to use it? And are drug development companies starting to do that now?
Yeah, I mean, there's a ton of investment in AI companies to help accelerate drug discovery. We'll have to see how much of it pans out. I think the best thing is I heard a talk from the chief scientific officer at Microsoft, who said that people who use AI will replace people who don't use AI. And it's sort of like when we were growing up in the 90s and people who use Google like would replace people who didn't use Google. Or, I hate to say it like so bluntly, but I had an aunt like who worked and made toothpaste and she didn't know how to use email in the 90s and got laid off, right? So unfortunately yeah.
I have a question kind of following on what you were talking about with the center of life science innovation, maybe shifting as a result of this. And are there other regions and countries that are investing in that pipeline of young and in the research and the researchers of the future?
Yeah, I mean, so it used to be, for example, in Europe, if you wanted to have a faculty position in Europe, you used to have to come to the United States to do what's called a postdoctoral fellowship. So you would train in a laboratory in the US and then you would go back with amazing credentials and stuff like that. Even when I lived in Basel in the mid 2010s, like 2014 and so on, that was already starting to not be the case because there were great labs already in Europe where people didn't from Europe have to come to the US to get the training and then go back. So that could accelerate. And then right now, I do have to say China also the government is investing incredibly a lot of money there. And they used to not be considered an innovation powerhouse in the life sciences, but now increasingly are making molecules faster and even cheaper and more creatively. And so that is we personally could be, how would I say it? I don't want to get too political, but we could be enabling who we're trying not to enable right here.
Could you see a scenario where the FDA is willing to accept Chinese generated clinical data?
No, that's already been tested. So maybe 5+ years ago, there were a bunch of US VCs that said, oh, we're just going to go get a molecule in China. It's approved in China and then get it approved cheaply in the US. And the US FDA said, no, you have to have US patients treated on those trials. So that would never happen, at least in the current environment. But you do see a lot of VCs going to China, buying a molecule, and then bringing it back to test in the United States and globally.
I have a question from the chat, which I think is along these lines. Do you have advice for students seeking to become research scientists but have been impacted by the funding cuts and are discouraged?
Yeah, so one of the themes of the book is determination and resilience. And so there's many setbacks in the sciences, and unfortunately, this might be one of them. But if you're really dedicated and committed, I would try to find a way to continue to pursue that dream. Hopefully there'll be other places that people could work or do internships or learn. And so this should not be a discouragement to enter the field of life sciences or drug discovery.
William, I have a question. So in terms of like affordability and getting these great drugs to people who need them, I understand sometimes gene therapy can be really expensive, and a lot of times, insurance isn't going to cover these kind of things. What are the things that I don't know, people in the industry do to try and make these drugs more affordable or available to those who need them?
Yeah, it's a great question. And I think that's why pharma really gets a bad rap on the drug pricing. Yeah, I mean, access, everyone agrees that access is really important. Question is, how is that possible? I do think, again, pharma advertising, if we could cut a large fraction or even eliminate pharma advertising and then just use that to really try to improve access to patients, that would be a start. As I mentioned, I think up to $30 billion is spent on advertising, actually. But when you watch the Super Bowl, like how many ads do you need to see on like some disease that you probably don't even have? Or how many times do you need to hear the jingle about Jardiance or whatever? It's like
But you do recognize the existence of very generous patient assistance programs that a lot of drug manufacturers have to try to get drugs in the hands of patients, not only in terms of free units, but in copay assistance regimes.
Yes, that's true. It still could be improved.
Undoubtedly.
I have a question about the patent process. Can you say anything about the patent process and how it plays into this story? Are patents protecting innovators or are patents limiting innovation?
Okay, Jean, what is your background? Are you a lawyer or?
My background is just interested in how this
Okay.
happens. I'm a historian, so I don't know anything
Interesting.
about life sciences.
Yeah, no, it's interesting. I mean, patents can go either way with the public, but they are critically important for innovation in drug discovery and a lot of other areas as well. Because you are able to basically, how would I say, leverage your discovery in order to then be able to sell the product that you develop. Now, there are controversies. For example, genes used to be patented, but there was a case that went to the Supreme Court that basically said like genes are part of nature, and they're why should you be able to patent that? And then the Supreme Court actually struck it down. Then there are also parts about a pharma where they extend these patents and try to put on all these use patents and try to extend the life of patents and so on. But fundamentally, pharma companies and other innovative companies still need patents in order to be able to sell their products. What I would say is you guys probably know that the lifetime of patents is limited on drugs. And so a company actually has to reinvent itself every year, every time the molecules come off patent. So, for example, a large molecule has 13 years. It's 17 years, I think. 17 years of patent life. And then a small molecule has like a little bit shorter than that. Now, I did say before, right? Sometimes you can take 10 plus years to even develop the molecule from beginning to end. And so even though 17 years might seem a long time, it actually may not be that long. And then after a company has developed that, then if that molecule goes off patent, then if the company doesn't have anything else, they can go bankrupt. And that happens, or they can get bought by another company and so on. So it is an industry where you have to reinvent yourself all the time. And you can't just make like an iPhone that's iPhone 13, 14, 15, 16, 17, right? Like people won't, you can't just iterate like that.
I have a question.
Sure.
Yeah, I have actually two. One, I'm a physician, but I still have never really understood how a gene therapy, such as the splicer that you described, going into the genome to correct the genetic error. I don't understand how the tropism of that happens. How does it go to the right place, and how does it get incorporated into the genetic materials? Is it just by the nature of the molecule and that's unique enough to go to the right tissues?
Yeah, it's a great question. So basically, the risdiplam is an oral small molecule. So it's a pill that you take, and then it actually gets into all of your cells.
Okay.
And so it corrects the defect in the relevant cells.
Gotcha.
So that's an instance where it's a small molecule that gets in, and then it happens to be, if it's a neuron, then it does the right thing.
Okay.
For a gene editing molecule, like the one Casgevy I was talking about for sickle cell, that actually, you have to take the cells out of the human body, edit them in the laboratory, and then put them back in the patient.
Do an exchange, plasma exchange.
Sort of like a plasma exchange, yeah, but it's
or dialysis?
Yeah, it's not quite called that, but essentially you do have to take the cells out and then put them back in.
Okay.
And you're directly manipulating the cells that you want to manipulate.
Okay. Are
It's a
there
great
any?
question. Yeah,
I mean, it
yeah.
just strikes me that, somebody has a liver problem and the drug acts at the liver. I mean, is there anything particular, or is it like your drug, the response platinum, that goes to all the cells, but it just happens to work at the one that is sick?
No, so if it's a small molecule, it would go throughout the whole body, right? Like if it's like
Okay.
Paxlovid, it goes throughout the
Okay.
whole body, and then well, Paxlovid is an example that kills a viral target, so it wouldn't
Right.
hurt anything in the body that might
Right.
irritate, but then it kills the virus and stuff like that. But
Gotcha.
any small molecule will have the best penetration throughout the whole body. The other class of molecules are antibodies, and they have a different kind of pharmacokinetic and pharmacodynamic profile, and so on.
Yeah,
So
but they have a target that they would, because they're of their conformation. That
correct.
would be specific. Yeah.
I just want to make everyone aware of the time. Not sure why I'm echoing. But it's 7:58. If you want to take a couple more questions, I want to be mindful of everyone. But yes, if anyone has one or two more questions, I believe we can take.
Hi, hi, William. Thank you so much for tonight's Zoom and for driving change in the medical landscape. It's great that we have this opportunity to hear from you. And, at the beginning of this Zoom, you mentioned that you see cancer doubling by 2050. And I'd love to ask you more about that, why you think that is, and why cancer treatments are still so difficult for us to figure out.
Yeah, so the epidemiology would say by 2050, the number of cases of cancer deaths is expected to increase by 91%, so almost double. So I think it's it's the increasing population. It's potentially more toxins in the environment. There is, like, for example, right now an increase in young people getting colon cancer for unknown reasons. Some of you may have heard about that, but like 20, 30-year-olds getting metastatic colon cancer. And so there are cancers that are still on the rise, unfortunately. And it's probably a mix of environment and genetics. Now, the reason cancer medicines are so hard to figure out is because you have to figure out a medicine that is killing your own body. I mean, the cancer cells come from your own body. So you're having to figure out how to kill those cancer cells, but not harm the normal body. And then cancer cells, their main goal is to survive. And so they like to mutate and continue to survive. And unfortunately, if you want to get mathematical about it, like a patient with metastatic disease has 10 to the 10th cancer cells already. So even if you get 99.9% killing, you still have like 1 million cells left, and then they can continue to populate and come back and make the cancer grow again. So there's just a lot of logistical, like, nature kind of issues in terms of tackling cancer. But we're making a lot of improvements. When I was a medical oncology fellow in 2000, the overall survival of lung cancer patients was less than a year. But with the drugs like osimertinib, the average survival of that subset of patients is 3 years. I know it's still not long enough, but there's now instances with immunotherapy, in melanoma, where patients are living 10 years beyond. So there's definitely progress, and we need to make more. So I know it's late. It's also 8 o'clock, at least for the people on the East Coast. So we should probably wrap it up. And I really appreciate all of you guys joining. It's really great to see a lot of familiar faces. And obviously, you can contact me individually if you have any particular questions.